Hello students, I hope all of you doing great and amazing and doing amazingly well in life. This is Basavaraj Sir, your biology master students. Welcome to Vedantu NEET English. Now students, today's class is going to be very conceptual, and if you are that student who does not understand what exactly is meiosis, then this is the class for you students. Because after this class, you will not only know every single concept, right? You will know the entire concept, but you will also be in such a good condition you can also teach to your friends also. That is my guarantee from today's students. Okay, students, if I'm properly audible and if I'm properly visible to all of you, can I quickly get a fire emoji in the chat to let me know, yes sir, I am audible and visible to all of you students. Hello. Good evening students. Good evening all of you students. Good evening all of you. Good evening students. Good evening. Now students, in today's class we'll be doing theory, right? We will be solving PYQs at the end of the session. So if you want to check your knowledge of whatever you are learning in today's class, stay tuned till the very end if you want to solve questions. But if you do not want to solve question, then also you should stay. Okay, then also you should stay for the end of the class, students. Okay, so students, before I start, all of you quickly mark your attendance by smashing that like button, all of you. So I want one of you to keep a reminder, right? I want one of you to be a monitor in the class. Keep reminding everyone to smash your like button. Okay. And students, before we start again, let me tell you students, we have finished the initial cell cycle. We have finished the M phase. Significance of mitosis is done. So if you have not watched that lecture, please watch mitosis before, because meiosis is very much related to mitosis. There is a wide connection, students. Okay, students, if you are aware that, right, we are coming to your city. So if you are from Hyderabad, Chennai, Telangana, anywhere, Bangalore, let us know in the comment section or let us know in the form. Then in the description there is a form. Click on that and you can see your where do you want us to come, because this weekend we will be coming to your city to meet all of you students. Okay, now students, now when we talk about meiosis, the first thing which needs to come to all of your mind is where does it happen and when does it happen. So meiosis is a very conceptual process. It happens in the germ cells. Students, can anyone give me the example? Can anyone give me an example of germ cell? So meiosis takes place in the germ cells. In the germ cells, students. It takes place in the germ cells. There are a few examples, students. There are a few examples such as spermatogonia and oogonia. The spermatogonia and the oogonia are the germ cells. They are the germ cells, all of you. Okay. Now, students, a question to all of you again. These germ cells, right? These germ cells, are they haploid or are they diploid in nature? Quickly in the chat right now. All of these germ cells are diploid in nature, diploid in nature. So when do you think meiosis takes place? Meiosis takes place in these germ cells during gamete formation. This spermatogonia will eventually turn into sperm. This oogonia will turn into egg. Now students, this spermatogonia, so this sperm, spermatozoa and this egg, they are haploid in nature. So students, the process which happens here between diploid to haploid conversion, this entire thing is possible because of meiosis. So students, a question to all of you. During meiosis, is there a change in chromosome number? Yes. In meiosis, students, in meiosis, there is always, remember, a diploid cell will become haploid cell. There is a change in set of chromosome. There is a decrease in entire set of chromosome undergoes decrease here. A diploid cell will become haploid cell, students, here. As you can see, students, here, a diploid cell will become haploid. So it happens in the case of germ cells where a diploid cell becomes haploid right now. Students, is that clear to all of you, students? Is this clear to all of you? Students, any doubt here? Quickly in the chat, students, quickly in the chat. So all of you know the location, all of you know what exactly happens. It happens in the case of germ cells, as well as there is a change in the ploidy here, change in the ploidy. Okay, now students, now I want all of you to understand what exactly happens in the case of meiosis, right? What exactly happens in the case of meiosis? The entire meiosis can be broadly classified into three categories. All of you write down. Entire meiosis is broadly classified into three categories. Okay, the entire meiosis is broadly classified into three categories. The first one is called as meiosis 1. Then we have meiosis 2 here. Meiosis 1, meiosis 2. So we have meiosis 1 and we have meiosis 2. Now, between meiosis 1 and meiosis 2, there is a very short-lived one more phase called as interkinase. Okay. Or interkinases. Interkinases. So the entire meiosis can be divided into meiosis 1, interkinases, and meiosis 2. Students, apart from this, in meiosis, I want all of you to remember, right? This entire thing is here. But apart from this, right, please remember this actually is the M phase, right? Now, I hope all of you remember. I hope all of you remember, students. When we say meiosis or mitosis, generally speaking, there are two categories. One is your M phase, right? One is your M phase, which is right here. This is your entire M phase, right? M phase. The other phase is always, students, the other phase is always the interphase. Okay, interphase here, students. Interphase. In this interphase, can anyone tell me what are the three stages here, students? Anyone? What are the three stages here, students? Anyone who can tell me? In this interphase, there are three stages. There is G1, S, G2. This M phase is this part here. Meiosis 1, meiosis 2, and interkinases. Interkinases. This is how it is actually divided. This is how it is actually divided. Okay. So meiosis can be divided into interphase, M phase, G1S, G2, meiosis 1, interkinases, and meiosis, students, meiosis 2. Now I want all of you to understand, students. We'll write here only. This meiosis 1 is divided into PMAT. P M A T, right? This is nothing but prophase 1, metaphase 1, anaphase 1, and telophase 1, right here. Now, students, then we have interkinases. Then we have meiosis 2. Meiosis 2 is again divided into PMAT. Prophase 2, metaphase 2, anaphase 2, and telophase 2. So here we can see the broad classification. You can see the broadly here how meiosis is divided into interphase, M phase, meiosis 1, interkinases, meiosis 2. Now, students, I want all of you to understand this interkinases. This one, it is actually very similar to interphase. It is very similar to interphase. Here also we can see G1 and G2. but there is a S phase also here. Here we can see G1 S G2 here, but in this interkinesis there is, students, it's very important here, very important, very important. Okay, in the case of your interkinesis, no DNA replication. So please remember, students, in the case of your interkinesis, here also we can find G1 as G2, but there is no DNA replication. DNA replication does not happen. The only time DNA replication will ever happen, the only time is S phase. Students, apart from that, it does not happen at all. Never possible that DNA replication can happen anywhere else. Okay, students, I hope, yes, Cash Army is here. Okay, now students, I want all of you to understand. Here I told you, in the case of meiosis, this diploid to haploid conversion happens, right? Diploid to haploid conversion happens. When this conversion is happening, right, when does it actually happen? Here, students, here. The conversion diploid to haploid conversion actually happens in the case of meiosis 1. So that is why, students, that is why meiosis 1 is also called as, students, it is also called as actual reduction division. That is meiosis 1. Now, students, but in the case of meiosis 2, do you see any further reduction? Do you, students, do you see that in meiosis 2, will there be a, let's say, n will become half n? Students, is it possible? 12th is going to be at 8 p.m. 11th is 6 p.m. generally. Okay, Plant Kingdom is coming up next week. Okay, Plant Kingdom is coming up next week. Don't worry, students. Do you think it happens, students, here? In meiosis 2, the n will become n by 2? Never. Students, please remember, it does not happen at all. So in the case of meiosis, the n to n conversion happens always. One cell will divide into two cells here. That's all happens here. But in the case of your meiosis 1, this one cell will divide into two cells like this. So diploid cell becomes haploid. Here haploid to haploid conversion happens. So students, can I tell meiosis 2 is similar to mitosis? Yes or no, students? All of you, yes or no? So technically speaking, students, it is actually equational division itself, just like mitosis. So students, all of you watching the lecture, students, right now, if you understand what exactly is mitosis from the previous lecture, then this part is done already, actually done. All of you know meiosis 2. So meiosis 2 is technically over, considering we already finished the entire mitosis where the reduction division happens. So the main agenda for today is to understand, to all of you, to properly get the brain drilled, is the meiosis 1. Okay, students. So with that being said, shall we start, students? Shall we start? Okay. Yes, students, ma'am is taking molecular basis because I will take some other time. Okay. Now, students, can we start a chapter right now? Is the overview clear to all of you? This is the overview of this chapter, right? Meiosis, interphase, I'll not be teaching you because interphase is again same here. This is what we'll be focusing today. This is what we'll be focusing. Now, students, I want all of you to tell me honestly in the chat right now, how many of you feel meiosis is difficult to understand? Sir, I cannot understand what is leptotin, zygotin, pachytine, diplotin, diakinesis. I do not understand what is prophase 2, prophase 1, metaphase 1, anaphase 1, telophase 1. You do not understand. Quickly in the chat, students, quickly in the chat. How many of you do not understand, students? How many of you do not understand that? Because this is very critical. It's very conceptual. You cannot by heart any of it, unfortunately, students. You cannot by heart it. So shall we start? Okay. Students, before I go ahead, I want all of you to realize that what is the necessity, what is the need for meiosis? Okay. First, I'll make you understand, is meiosis even required? Can we survive without meiosis? That is first understand, students. Okay, I'll give you a quick scenario right now. Students. Quick scenario. Okay. Now, I want all of you to understand, right? Students, here we have a male, here we have a female. Okay. Nice hair we'll give. Male and female. We'll give color hair. Wait, students. We'll give which color, students? We'll give orange color hair. Okay. Male and female. Right. Male is producing the sperm here. Female is producing egg. Male and the female are diploid in nature. Diploid in nature, students. Right. The sperm is going to be haploid. Haploid. Why? Because of meiosis. And when they fuse together, we get the zygote, which is going to be diploid in nature. So diploid here, haploid. This zygote will develop into female, male again. Now, students, now, I want all of you to understand, students. I want all of you to understand what will happen. Right? What will happen if there is no meiosis? Okay, I'll give you an idea. Let's say meiosis is not there. If meiosis is not there, then what happens? Okay, this person is diploid. Then if there is no meiosis and only there is mitosis, if there is only mitosis, then what happens? This sperm is going to be diploid itself, egg is going to be diploid, the zygote is going to be foreign then. Foreign, yes or no? If there is no meiosis. Similarly, there will be one more zygote formation here. Let's say we have one more zygote here from one more parents. This zygote is again foreign condition. Yes or no? Let's say this develops into a male. This develops into a female now. Female here. Here, this person is foreign condition. This person is foreign condition. Now again, gametes will be produced. Gametes are going to be foreign again. And this new zygote is going to be 8 in condition. So do you realize, students, if there is no meiosis, the ratio will never match and will become aliens. Next generation will not even look like human. The next generation will not even look human if there is no proper meiosis. So meiosis becomes very, very important for the survival of the organisms. Yes, sir, students? Did all of you understand, students? Did all of you understand why exactly we need the meiosis? Without meiosis, life cannot exist at all. Students. Now here we see, students, meiosis has meiosis 1 and we have meiosis 2. Meiosis 1 and meiosis 2. Now here we'll be focusing on, first of all, we'll be focusing on prophase 1. Students, shall we start? Students, shall we start? All of you. Students, shall we start meiosis 1 right now? Shall we start meiosis 1? In meiosis 1, the first topic is called as prophase 1. Okay. In prophase 1, we actually have, under prophase 1, we have 1, 2, 3, 4, 5. So under prophase 1, we have 5 different stages. We have 5 different stages under prophase 1 itself. Now it's called as leptotene, zygotene, pachytene, diplotene, diakinesis. Now how do you remember this? Small trick here. Let zebra play during the day. Here L, Z, P, D and D. So that right here is your prophase 1, leptotene, zygotene, pachytene, diplotene, diakinesis. Those are the 5 different sub-stages of prophase 1. Here, of this prophase 1, there are 5 stages of this prophase 1 right here, students. Prophase 1. Okay. Students, repeat after me, students. I want everyone to type this in the comment section right now. All of you type this. Let zebra play during the day. Okay. Leptotene, zygotene, pachytene, diplotene, diakinesis. Okay. Leptotene, zygotene, pachytene, diplotene, diakinesis. This order is very important. This order is very, very important. Order is very important. There has been PYQs based on the order also. There has been PYQs. Okay. Now, students. Now, first we understand leptotene, students. First, we need to understand what is leptotene, right, student? Leptotene. Now, students, I hope all of you remember before the start of prophase, before the start of prophase, the DNA is present as chromatin. All of you, see students, this prophase is technically after your interphase, and in interphase the DNA is present as chromatin. So in the case of prophase 1, what happens? In the case of prophase 1, the first stage leptotin, what happens students? Chromosome gradually become visible under light microscope. So this is very important, students. This is the main point here. This is the only and only the main point, students. Main point here. Okay. This is the main point right now, students. Okay. Students, if you want to understand, if you really want to understand plant kingdom, learn this chapter first. Without learning this chapter, you cannot understand plant kingdom. You cannot understand at all. This chapter is more important than plant kingdom. Okay. Students, under light microscope here. So this is the only point you have to remember here. Point number 1. So I will write all the points with pink color here, so we can see it. So this is the only point we have to remember here, students. Only point you have to remember. Okay. Leptotin is over. Leptotin is over actually. Okay. Now, second point, second stage is called as zygotine, students. Second stage is called as zygotine. Now, I want all of you to understand what exactly happens in the case of zygotine. Now, students, in your class, have you ever made a group project? Students, have you made projects any time? In your school, in your class, have you made projects? Okay. If you have made projects, usually what they do is they ask you to make a group of 2, a group of 3, or group of 4 also. But generally, when I take a class, I say group of 2. Now, let's say you make a group of 2 right now, right? Group of 2. Now, do you choose any random person to be your project partner, or do you choose your best friend in the class? Students, tell me, students, in the chat. All of you. All students. All of you students. Do you choose any random person, or do you choose your best friend? You choose your best friend, right? Yes. So inside a cell also right now, there will be a selection process. Inside a cell, there will be a selection process right now, selection process. Now, what do I mean by that? In the selection process, what is happening is that, right, there is in total of, remember students, do you remember? In total of, there are 46 chromosomes in a cell, right? We also call it as 23 pair. Yes or no, students? We call it 23 pair. Yes, we call it 23 pair. So here also what happens is that all the chromosomes will start to pair together, and these chromosomes will start pairing together. They are called as homologous chromosomes because they look alike and they also have the same genes on the same loci, right? Basically, they are copies of each other, best friends. So two best friend chromosomes called as homologous chromosomes, two best friend chromosomes called as homologous chromosomes, they will start coming together. Now let's see, students, when two chromosomes are trying to close together, that process is called as what? The process is called as synapses. Synapses. That is point number 1, right, students? That is point number 1 here. That is, chromosomes start pairing together. Okay. Which chromosome, students? Homologous chromosomes. So homologous chromosomes will start to pair together. The process of pairing together is called as what, students? What do you call that, all of you in the chat? The pairing of chromosomes is called as synapses. It is called as synapses. Synapses. Let's draw two chromosomes, students, here. Let's draw two chromosomes here. Okay. This is one chromosome, students, here. And then we have second chromosome here. The two chromosomes, students, the two chromosomes will start pairing together. Something like this. They're pairing together. Now, this is called as synapses, students. This is called as synapses. Okay, this is called as what? Synapses. Now, students, what happens? Between the two chromosomes, right? Between the two chromosomes, there is, oops, what happened, students? What happened? Between the two chromosomes, right? There is a complex formation right now, students. Students, voice break, I'll clear it. Next session, I'll get it rectified. Don't worry, students. Now, between the two chromosomes, right, there is a complex formation right now. Students, all of you are able to see? Students, all of you are able to see the complex here? Okay. Now, students, I will call this complex right here as synaptonemal complex. Done. So, students, that is the point number 2 right now, students. Okay. That is the point number 2 right now. Okay. That is called as point number 2 here. Okay. So, point number 1 was synapses. Point number 2 is synaptonemal complex formation. Now, students, in this particular stage of your cell cycle, cell division, right, this stage is also called as tetrad, or it is also called as anyone, students? It is also called as tetrad or diad. Okay, so this stage, students, is called as your bivalent to tetrad, sorry, not tetrad, not diad. Diad also comes later on. This stage right here, students, this stage right here, it is called as bivalent or tetrad. Now, students, you will ask me, sir, why do I call it bivalent? Why do I call it tetrad? Very simple, students. Now, I want all of you tell me, students, I want all of you tell me, how many chromosomes do you see here? Students, how many chromosomes do you see here? How many chromosomes? One here, one here. So, bivalent. That is why I will call it as bivalent. Okay. Then, students, how many chromatids do I see here? Chromatids. How many chromatids? All of you count with me. Here we have 1, 2, 3 and 4. I see 4 chromatids here. So I can also call it as tetrad. I can also call it as tetrad right now. So it can be called as bivalent or tetrad. Stage is around here, and that right there is point number 3. So 3 points with respect to zygotine. Synapses formation, 2 chromosomes coming together, complex formation, that is point number 2, and tetrad or bivalent formation, that is point number 3. Any doubt, students? Any doubt here? Can I tell zygotine is done? Students, can I tell zygotine is done right now? Done zygotine? All of you in the chat? Okay, students, zygotine is done. Now, you can see, students, this is how the formation happens. This is how the formation happens. Okay. Now, students, now we come to pachytene. Now, in pachytene, there are multiple points which all of you need to remember. Multiple points are there. Multiple. Okay. The first point is that, students, remember the zygotine, the bivalent and tetrad stage. The bivalent and tetrad stage becomes much more clearly visible. My words, students, very important here. Bivalent chromosomes clearly appear as tetrad here. So the tetrad will look very clear right now. Unlike my voice. Okay. Unlike my voice, it becomes very clear right now. Okay. So that right here is the point number 1, students. That right here is the point number 1. The bivalent or the tetrad becomes very clearly visible, students. Clearly visible. Okay. Now, students, apart from that, there is something called as crossing over. Students, how many of you know what is crossing over? Or if you want, right now, I will draw and show you what is crossing over, students. What is crossing over? Students, do you want to see with the help of a diagram what exactly is crossing over? Because I'll give you a definition first of all. I'll give you a definition, right on the definition, students. What exactly is crossing over? Then I will show you, students. Crossing over is nothing but exchange of genetic material between non-sister chromatids of homologous chromosomes. This right here is the definition of crossing over. Now, students, before I show you the diagrammatically, I want all of you to understand, if I have two chromosomes here, right? Two chromosomes here, they are homologous chromosomes. They are the homologous chromosomes. The meaning of homologous chromosomes is that these two chromosomes are identical to each other. Now here, this and this, if I take one from here and one from here, these two chromatids are called as sister. One from here and one from here, they are called as brother, not brother, non-sister. They could have called it brother, but they're not called brother. They're called as non-sister. They are called as non-sister. Okay, they are called as non-sister. Non-sister. This right here is, these two are called as sisters because they belong to the same family, that is same chromosome. One from here is called as non-sister. It is called as non-sister right now. Let me just draw and show you, students. Let me just draw and show you right now. We'll use the same color. We'll use the same color here to understand crossing over. Okay. Now let's understand crossing over, students. Okay. Now, students, let's just take first chromosome here. This is the first chromosome. This is the second chromosome right here, students. Now, between them, what do we have? Between them, we have the synaptonemal complex. Okay. Now, what happens, students? Now, what happens? Now, at one point, they will start to cross over. Now, as you can see, they are trying to cross over right now. Now, during this process, there can be formation. This is called as what? This right here is called as site of crossing over. This right here is called as site of crossing over. In this site of crossing over, students, in this site of crossing over right here, right? This point here. Here we can form something called as recombination nodule formation. There is also recombination nodule formation here. So at the site of, students, at the site of crossing over right now, right? At the site of crossing over, we can see something called as recombination nodule. Nodule is like one round color structure there, right? Round color structure, which basically helps in the crossing over process. Entire crossing over process, it helps. Okay, that is called recombination nodule. It has a lot of proteins and everything. This right here is the crossing over site here. Now, students, here there is also enzyme involved also. The enzyme here involved is called as recombinase enzyme. Okay, the enzyme involved in this process is called as recombinase enzyme is present here. Now, students, what happens? Now, what happens after this? I'll just draw from here, students. I'll just draw from here. Now, what happens, students, after crossing over? Now, after crossing over, students, here we can see the synaptonemal complex also is here. This right here is the enzyme. Enzyme involved here. And this is called as recombination nodule formation here. We have the recombination nodule formation also here. Okay. Now let's just draw this last part here, students. What exactly happens right now? Okay. Now here we have the first chromosome. Now look at the chromatid. Look at the chromatid right now. Look at the chromatid right now. This is called as crossing over, students. This right here is called as crossing over. Okay. So centromere is right here. Centrosome is a organelle. Centrosome is the organelle, centromere is this middle part here. Students, this is called as crossing over. Okay. Now students, I will teach you how many points which you have to remember here. The first point, students, all of you, the first point remember here is bivalent chromosomes clearly appear as tetrad here. Second point is the site at the site of crossing. Second point is the crossing over. Second point is the crossing over here. Definition I've given you. The second point definition also I've given you. Crossing over. There is basically exchange of genetic material between non-cystic chromatids are exchanging, students. That is the second point right now here. Second point. You can see here also, students, second point. Now what is third point, students? Third point is going to be the formation of recombination nodule. Formation of recombination nodule is the third point here. Fourth point is the recombinase enzyme here. Recombinase enzyme, students. So with that, we have 4 different points for your packeting. Is that done, students? Is that done, all of you in the chat? Packeting is done? All of you. Packeting is done. Students, I'll repeat again. First, bivalent clearly appear as tetrads. Point number 1. Point number 2 is going to be crossing over. Point number 3 is going to be recombination nodule formation. Point number 4 is going to, students. Point number 4 is going to be recombinase enzyme. With that, it is done. So students, why so much of love suddenly? Huh? Why so much of love? What do you want? All of you tell me, students. Do you want notes? I'll give you notes. Don't worry, students. Don't cry. Why so much of love suddenly, students? So much of affection you should not show suddenly. It becomes overwhelming. Overwhelming it becomes. Okay, so much of love suddenly. Okay, don't worry, you'll get the notes. Do not worry, I will give you all the notes. Do not worry, I will send you all the notes and I will finish the syllabus. Okay, don't so much muska and all so much. Okay. So once again, recombination, nothing but is crossing over here. That is nothing but crossing over. Students. See, let me show you one more thing. Students. Can you see? Here we have 2 homologous chromosomes here. This is called as the site of crossing over here. Site of crossing over. Here we can see recombination nodules also here. Okay. Then you can, students. Then you can see here, students. Then you can see, students. Here. Then you can see here. Okay. So you can see here, this chromatid one piece is on this side. This one is on this side. They're basically doing exchange offer here. Exchange offer. Now that is, these are basically, these two are recombinants right now. These two are non-recombinants. These two are non-recombinants. These two are recombinants right now. Recombinants and non-recombinant, students. Non-recombinants. Okay. Now students, now we come to diplotin, students. Diplotin. I will draw and show you. Please follow with me. Follow with me, students. Students, all of you making the notes. If you're not making notes, do not worry. I'll give you the notes. Now, students, this is what I have, right? This is what I have right now. One has come here. One family member has got married and they've come here. Okay. One second, which color was it? Okay. Now, students, the first point in the case of diplotin. The first point, students, the first point in the case of diplotin is that, right? The first point in the case of diplotin is that, students, can anyone tell me what is this middle part here? Anyone? This middle part here? What is this part here? This part? That is called as synaptonemal complex, students. Yes or no? It is called as synaptonemal complex. This complex will now disappear. That is the first point here. That is dissolution of synaptonemal complex, point number 1. Okay. Now, students, after that, these two chromosomes will start to go apart. They're like, marriage is over. I will go to my house. You go to your house now. So both chromosomes will start to separate from each other. They will separate except at one point. These two chromosomes will start to separate from each other except at the point of crossing over. And here we can see a X-shaped mark here. This X-shaped mark right here is called as, students, it is called as chiasmata. It is called as chiasmata, students. So the two chromosomes will go apart from each other except at the X mark. The X mark is called as chiasmata. So if I ask you a question right now in the chat, if I ask you, students, if I ask you a question, okay, if I make you ask you a question right now, chiasmata formation happens in which stage of prophase 1? What is your answer, students? What is your answer? All of you students in the chat. Yes, border me is slaying today, slaying. Students, all of you in the chat, quickly. Chiasmata formation happens in which stage? It happens in which stage of your prophase 1? It happens in the pachytene. So please remember that. So not pachytene, it happens in your diplotin. It happens in diplotin. Okay, chiasmata formation happens in diplotin right now. Diplotin, right? Chiasmata formation. The X-shaped mark here. Okay, students. Now we come to the last part called a diakinase, students. We come to the last part called as diakinases. Okay. Now, students, this is the X-shaped mark, no? X-shaped mark. Now, what happens is that this X-shaped mark also will start to terminate. That is called as termination of chiasmata. This X-shaped mark is there, right? That also will separate out. Now, that is called as termination of chiasmata. That is point number 1 right now. Students, here we had 3 points, right? First point was synaptic complex. Second point is chiasmata formation. 2 points here mainly. Okay. Now, students, see, okay. Then diakinesis, termination of chiasmata, point number 1 here. Chromosomes fully condensed, point number 2 here. Spindle mitotic assemble, that is the spindle fibers will start to arise right now. Spindle fibers, yes or no, students? Spindle fibers will arise right now. Okay. Nucleolus disappears, nuclear envelope disappears, represent transition metaphase. Basically, every other organelle is disappearing right now. So everything organelle disappears. That is point number 4. And with that, prophase 1 is completed. You right now finished the most difficult part of your entire class 11th. Done. So students, I will give you right now an entire breakdown of entire prophase 1. Okay. Students, see this right now. Okay. Now I want all of you to remember, students. I hope all of you remember. We have meiosis, we have interphase, M phase. Meiosis 1, interkinase, meiosis 2. In meiosis 1, we have prophase 1, metaphase 1, anaphase 1, telophase 1, all different phases here. Here, prophase 1, students. Prophase 1 has 5 different stages here. Prophase 1, we can see leptotene, zygotene, pachytene, diplotin, diakinases under prophase 1. Now, first, leptotene is what? Chromosomes gradually become visible under light microscope. Point number 1. In zygotene, students, what happens? There is synapsis. What is synapsis? 2 chromosomes coming together, homologous chromosomes, synapsis. Now, once the chromosomes come together, there is a complex formation, right? Synaptonemal complex formation. Okay, done. Now, after that, this stage is also called as bivalent or tetrad. Point number 3. Then, students, then we have the pachytene right now. In pachytene, we have first point here. That is, the chromosomes clearly appear as tetrad right now. The first point. Second point is crossing over. Here's the definition for crossing over. Point number 3 is recombination nodules at the site of crossing over. Point number 4 is the recombinase enzyme present here, students. Present here. Now, students, then we come to diplotin, students. In diplotin, what happens? The first point is synaptonemal complex disappears. Then there is an X-shaped mark formation called as chiasmata. Students, remember this point. Recombined homologous chromosomes separate from each other at all sites except at the site of crossing over. Except at the site of crossing over, they will separate from each other. And this X-shaped mark is called as chiasmata formation. 2 points here. Then we have diakinase, students. In diakinases, termination of chiasmata. First point, separate out. Yes. Then we have chromosomes condensed fully right now. Fully condensed. Mitotic spindle assembles, that is spindle fiber formation, and every other organelle disappears. And that is the end of your prophase 1, students. Prophase 1. Any doubt till now, students? Any doubt till now? Students, any doubt till now? Yes, last class. This is my last class. Not my last class, this is the last class of this chapter. Students, any doubt? Any doubt in the entire prophase 1? Now students, now what do we do, students? Now what do we do? Now we have metaphase 1, students. Now we have something called as metaphase. Students, I am trying to teach you as slow as possible, because if I rush anywhere, you will not understand the concept. Okay, now students, now Sir, won't bivalent complex dissolve and split again to the homologous chromosomes? No, they will say, see, the complex is gone, they will still remain together. Students, understand, once the two homologous chromosomes are together, they will not separate out until anaphase. Until anaphase, the two homologous chromosomes will not separate out. They'll still be together, they're paired together. Until someone is pulling them apart, which happens in anaphase. Until then, they're together. Okay? Okay, students. Students, I hope all of you remember, right? I hope all of you remember, students. Right? Here, this diploidine stage can last for a lot of years actually. If you remember, students, if you remember the oogenesis, I told you that, remember, in oogenesis, in entire oogenesis, in exactly prophase 1 diploidine stage, the cell was arrested until puberty. Do you remember? At puberty, we got the secondary oocyte. Do you remember? The primary oocyte was completely suspended. Diploidine itself, student. That is diploidine itself. Okay, diploidine, students. Diploidine itself. Okay. Students. At diploidine itself, it was suspended. Remember, in oogenesis, I told you, in oogenesis. Now, students. Now, in metaphase, what happens? In metaphase, the bivalent will align on the equatorial plate. Now, students, if you remember, right? I hope all of you remember. If not, it'll be problem. In the case of, let me give you a side-by-side comparison right now. Okay. Now, this was in the case of your mitosis. This is in the case of meiosis. In the case of mitosis, what happened, students? In the case of mitosis, if you remember, all the chromosomes were aligned like this. Yes or no? Students, do all of you remember? In mitosis, yes or no? Because it is a very complex process, students. Yes, quiz is there. Quiz is not there. I'll ask you questions. There are PYQs there after the class. Okay. Now, students, in the case of meiosis, what happens? In the case of meiosis, here we have only one plate. Metaphasic plate is only one. But in the case of meiosis, students, please understand, in the case of meiosis, there will be two plate formation like this. Because we have the homologous chromosomes right now. So, in the case of metaphase 1, we will see two lines like this. This is the pair of chromosomes. This is the entire pair of chromosomes right now. Pair of chromosomes in the case of your meiosis right now. Now, students, here in the corner we can see the two centrioles. That is the entire centrosome right now. Now, from here, the spindle fiber will arise. Now, this is how it happens in the case of what happens in the case of meiosis. So, in the case of meiosis, this is what the metaphase looks like. Okay. Now, this is your metaphase 1. Bivalent align at the equatorial plate, microtubules of spindle attached to the pair of homologous chromosomes. Now, students, all of you tell me what will happen if I hold this part and I hold this part, and if I pull it on both sides, what will happen? Will the centromere split? No. So, can I tell, if I start pulling from both the sides, will this entire set will go up, this entire set will go down? All of you agree with me, students? Yes or no in the chat quickly. This entire set is going to go up, this entire set is going to go down. That is, in total here I have 46 chromosomes, 23. I have in total 46 chromosomes, and here on the top I have 23, on the bottom I have 23. Yes or no, students? Yes or no? So if I pull this up, if I pull this down, that is what happens in the case of anaphase 1. So in the case of anaphase 1, what is happening? In the case of anaphase 1, homologous chromosomes separate. Sister chromatids remain associated. Associated means joined here. Joined. They are together itself. That is, these two sister chromatids will be together. This entire set is going to go up, this entire set is going to go down, and there will be separation of homologous chromosomes will separate out right now. Separate out. Okay. Now, students, now we come to telophase. Students, very. This is what is happening, students. This is what is happening in the case of anaphase. Anaphase. This entire set is being pulled to this side, entire set is being pulled this side. Okay. So, out of 46 chromosomes, 23 are going on top, 23 are going on top, 23 are going at the bottom. Students, are you noticing right now, students? Are you noticing there is a haploid condition is being formed right now? Do you notice? The haploid condition is being formed right now. From a diploid condition, we are splitting the chromosomes, homologous chromosomes, after crossing over into haploid condition. Now, this is what happens in the case of telophase. In telophase, the chromosomes gradually cluster, nucleolus, nuclear membrane reappears. Then we have everything is reappearing here. Chromosomes are on the opposite end here. Chromosomes are at the opposite end right now, students. Okay. So here we end up getting two cells at the end. This right here is called as your dyad of cells. So from anaphase, telophase takes place. Then we have cytokinesis, and we end up getting two cells. We end up getting two cells eventually. So this right now here, we had 46 chromosomes right now. Yes or no? This entire 46 chromosomes got divided into 23 on the top, 23 at the bottom. Here, further division, 23 chromosomes here, 23 chromosomes here. We divide into two cells. 23 chromosomes here, 23 chromosomes here. So what actually happened here? We had a diploid cell. It now gave rise to two haploid cells. Here we had 46 chromosomes. We end up getting two cells which are 23 and 23. That is what happened in the case of telophase, students. That is what happened in the case of telophase. What do you mean? What are the remaining 46? 46 became 23. Simple as that. Simple math. We had 46 chromosomes in the entire cell. But exactly at here, you can see here, students. You can see here also. Exactly here we have 46 here. Total 46. 23 are aligned here. 23 are aligned here. This 23 is pulled this side. 23 are pulled that side. That is what is happening here in anaphase 1. Anaphase 1. Pulled apart. And we end up getting two haploid cells. Students, with that, your entire, this is the end of telophase 1 actually. This is the end of telophase. End of telophase, 46 chromosomes, 23 went this side and remaining 23 went this side. 23. Okay. Now, students, if that is clear to all of you, interkinesis are all of you, students. Interkinesis, I told you. In interkinesis, there is no DNA replication, at all. But there is something called as G1 and G2 will happen. Okay. Now, students, if you remember mitosis, this is easy for you. This is very easy for you, students. Very easy for you. Okay. Now, what happens, students? What happens? Prophase, all of you know. What is prophase? Nuclear membrane disappears. Chromosome becomes compact. That is compaction of chromosomes. That's all. What happens in metaphase, students? In metaphase, everything aligns in the center. Everything aligns in the center right now. Okay. Yes. The same thing will not happen in meiosis 2. Please remember. Okay. Everything aligns in the center right now. Center. Now anaphase students, anaphase 2, what happens? In the case of anaphase 2, the sister chromatids separate right now. See students, remember, anaphase 1, homologous chromosomes separate out, but anaphase 2 is like mitosis. So what happens here? Centromere splits. If this is one cell here, this is how the chromosomes were aligned here. The centromere is splitting right now, splitting. This half of will go this side, half will go that side, just like mitosis students, just like mitosis. Students, do all of you remember or not? Should I show you what exactly happened in mitosis? Students, do all of you remember or not? Tell me quickly in the chat, students. Students, I hope you remember this mitosis. I told you very well. Remember mitosis? I told you, students. See here. See, metaphase at the center, centromere, kinetochore, right? Anaphase, what happens, students? In anaphase, it split open. The sister chromatids, the centromere split open. This half went this side, this half went on top. The splitting of centromere happened. The same thing is happening in the case of meiosis 2. Same thing is happening in the case of meiosis 2 only. Okay. Same thing is happening in the case of meiosis 2 also. Anaphase 2. Telophase 2, what happens, students? Nuclear envelope and nucleolus reappears. Cytokinesis occurs. Formation of tetrad, that is 4 haploid cells happened right now. That is, one second, students, 1 second. Initially we had 2n condition. Initially we had 2n condition. We got 2 cells at the end. These cells divided n and n, n and n. Here to here is called as meiosis 1. Here to here is called as meiosis 2. Meiosis 1 is very important, but meiosis 2 is just like mitosis. Everything you learn in mitosis, same thing applies here also, students. Okay, same thing applies here. If you remember mitosis, meiosis 2 is same to same. Okay, if you don't remember, students, let me just show you. I think I closed it. Okay, so the same thing, whatever you learned in mitosis, is same thing is there in the meiosis 2. Okay, now students, I will teach you something very important right now, that is called as DNA content. Okay, if you learn the DNA content, this chapter is done. This chapter is done, students, if you learn DNA content. Shall we start, students? Shall we start? All of you? Students, here is your significance. Conservation of chromosome number. I told in the starting, remember, diploid condition is maintained. Genetic variability, evolution. Okay. Now, students, now we come to something called as DNA content. Okay. So, pro to ana, both meiosis and mitosis same? No. Meiosis 1 is very different. In meiosis 1, I'll give you a recap, students, before you go DNA content. Meiosis 1 is very different actually. Please remember, students, please remember. See, students, here. Meiosis 1, meiosis 2. Pro, meta, ana, telo. Pro, meta, ana, telo. Interkinase in the middle. Prophase 1 has 5 paths. 5 paths. Leptotene, zygotine, pacatine, diplotin, and diakinesis. Then we have metaphase 1, where this happens in the case of mitosis. This happens in the case of meiosis here. Alignment is there. So in the case of your anaphase 1, what happens? Anaphase 1, homologous chromosomes separate, not centromere. Anaphase 1. Telophase 1, we get end of it, we get dyad of cells. Then students, then we have meiosis 2. Meiosis 2 is similar to mitosis. Like prophase, what happens? Metaphase, only single plate here. Sister chromatids will separate out. They'll separate out, your students. Telophase, everything again reappears. Everything is reappearing there. That's all is your meiosis actually. Okay. See, anaphase, sister chromatids are separating out. Telophase, everything is reappearing as chromatin here. And we get dyad of cells. Now students, I'll teach you something called as genetic content or DNA content. Please learn this if you want to solve some good questions. When I say good question, let me show you what type of question sometimes. Okay. Students, see this question. Let's see how many of you can solve this question. It's a challenge to all of you. This right here is challenge to all of you, students. Challenge to all of you. How many of you can solve this question right now? How many of you can solve this question? A challenge for all of you. Students, if you want to solve such questions, you need to know the DNA content. Shall we start, students? Shall we start? Students, shall we start? See, students, I have so many questions. I have a lot of questions today. So we'll come for that. Okay. Now, students, let me teach you DNA content right now. Let me teach you DNA content. Ready, all of you? Ready for DNA content? Okay. Now, let's take a human cell. We'll be taking a human cell. Human cell which is diploid in nature, which has 46 chromosomes right now. We'll be taking the DNA content in the starting 40 picograms. Okay, done. Now, students, now let's take first things first column here, that is the stage. Okay, stage is done. Now, after stage, we will do number of chromosomes. Then we will do number of chromatids. Then we will do DNA content. Then we will do equation. Okay. Okay. Now, students, first we will solve for meiosis 1. Okay. First we will do for meiosis 1, right, students. First we will do for picogram. PG is picogram DNA content. Okay. Now, students, the first stage is your G1. Then we have S. Then we have G2. Yes or no, students? G1, S, G2. Then we have pro, meta, ana. Then we have the telo. Telo, I'll teach you per cell and per nuclei is going to be different here. Per cell and per nuclei is going to be different here. Okay. Now, students, initially we are starting with G1 here. Number of chromosomes is 46. Number of chromatid, students, every single chromosome in the starting has one chromatid. One chromatid, students. Yes or no? It looks something like this right now. So here also we will have 46. DNA content is 40 picogram. 2 and 2C condition in the starting. 2 and 2C condition starting, student. Okay. 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 Now, students, now in S phase, what happens? Number of chromosomes will remain the same, but the DNA content will double. Yes or no? So, number of chromatid, one chromatid is becoming two chromatid in every single chromosome. So, 46 will become what? 92 chromatids. Every single chromosome will have get double. Every single chromosome which had initially one chromatid, now we will have two chromatids right now. So, 40 will become 92. 40 will become 80. 2 and 2C will become 2 and 4C. 2 and 4C. Done. This was clear to all of you in the last class also. Okay. Same thing happens till here. Now, when we come to telophase, per cell, it is still the same. Per cell. Now, per nuclei, here is the main difference, students. Main difference when we come to nuclei. Understand, students, what happens per nuclei right now. In the case of meiosis 1, let's just go back here. Where is here? Okay. Now, students, let's say this is one entire cell right now before cytokinesis. Before cytokinesis, right here, students. Okay, let me just teach you from here only. Okay. Now, right now, it is in anaphase right now. Okay. Now, this right here is 2N4C right now per cell. 2N4C. This right here is 2N4C. It means that there are in total there are 46 chromosomes right now. Now this 46 chromosomes, half is going on the top and half is going on the bottom. That is 23 is going on the top right now and 23 at the bottom. So I have to divide right now, students. I have to divide this entire 2N4C, this entire cell, by half, because half of the chromosomes are going this side, half of the chromosomes is going this side, half of the DNA content is going this side, and remaining half of DNA content is going this side. So by the end of telophase, per nuclei, half of 2N is N, half of 4C is 2C. So N2C is present here and this is N2C right now. So per nuclei, right, per nuclei, N2C is here and N2C here. So 2 and 4C, that was the entire cell. Let's draw here also, students. Here we had entire cell is 2 and 4C, but when it is dividing per nuclei, this one nuclei, each nucleus, students, out of this 46 chromosomes, 23 will go here, 23 will go here. So right now, per nuclei, how many chromosomes, students? Tell me in the chat quickly. All of you, students, all of you. Per nuclei, how many chromosomes do I have right now? Per nuclei, I will have 23 here. 23 here per nuclei. How many chromatid, students? 46 chromatid, because every single chromosome still has 2 chromatid. Per nuclei, DNA content is become 40 here. And this 2 and 4 is going to become N2C. N2C. Right? So, we are right now ending with N2C condition per nuclei at telophase. N2C condition, students. N2C condition. N2C condition, everything becomes half right now. So this right now, from a diploid cell, from one diploid cell, did we get a haploid cell, students? All of you agree with me? From a diploid cell, we ended up getting a haploid cell. That is what happens in the case of meiosis 1. In the case of meiosis 1, that's what happens. Now, students, now we come to meiosis 2. Meiosis 2, students. Okay. Meiosis 2 right now. Understand, students. Understand. Okay. Now, here we have G1. G1 is there. Okay. In G1, number of chromosomes, students, from previously we had 23. Number of chromatids, 46. DNA content, 40 picogram, and N2C condition. Same thing is, students, repeating. Same thing is repeating right now. Now, students, here, do you think in S phase and in G2, will there be a change right now, students? Absolutely not. Because if you remember, I told you in bit after meiosis 1, in interkinase, there is no increase in DNA content. So it will remain the same itself. 23, 46, 40, N2C. It will remain the same, students. It will remain the same itself. It will not change, because I told you there is never change in DNA content anymore. In interkinase, there is never a change. Okay, never a change. Okay, students. Now, in metaphase, remains the same. In the case of anaphase, there is a change, students. Anaphase. So, students, if you remember the anaphase of your mitosis also, mitosis anaphase. Here, remember, students, if you remember in the case of anaphase also, mitosis, every single chromatid is acting like a chromosome temporarily. If you remember, So, 46 chromatids will become 46 chromosomes right now temporarily. This will be 46 itself, 46 itself. This will become 40, it will remain 40 itself, and this now, this right now will become 46 is 2 and 2C condition. Temporarily, students, temporarily in the case of anaphase, we have 2 and 2C condition. Students, I hope you remember in the case of anaphase, I told you, every single chromatid is acting like a chromosome. So temporarily there, in mitosis, it became 4N condition, if you remember, temporarily. So here, temporarily, it becomes 2 and 2C in anaphase. Okay? Now, right. Now in telophase again, per cell and per nuclei. Per cell remain the same. Now per nuclei, what happens? Students, understand. Per nuclei in the telophase 2, right? Telophase 2. All this is 2 to 2. Remember, students, if you remember here, right now, this entire cell, right, is 2 and 2C. All of you remember? This right here is 2 and 2C. This entire cell is 2 and 2C per cell. Now, students, if I tell you, right, if I tell you, out of this 2N, 1N is this side, 1N is this side, 1C is this side, and 1C is this side. That is how it is dividing, students. That is how it is dividing, students. Out of this 2 and 2C, this side is N, half C. This side we have N and C right now. Students. So that is per nuclei, students. So if you look at the final table, at this final line, here per nuclei, it will have 23 chromosomes, again haploid nature, restored. Everything will have one chromatid, obviously, because here the centromere splits here, 23. 40 will become 20, and 2 and 2C will become NC condition. So we are ending with NC. Condition. We do not counter quotient because it is happening after G1. So whatever is present after G1, right? Same thing is there in quotient also. Yes or no? So in the end, we have NC condition. Now let's look at the question what was given to all of us before. This was the question given to all of us, students. The question said, after meiosis 1, right, the resultant daughter cell will have, after meiosis 1. Okay, students, if you know the table, okay, we can just go back here, and after meiosis 1, we have N2C condition. Okay, we have N2C condition after meiosis 1. Okay, N2C, remember, students, remember. So after meiosis 1, we have N2C condition. Okay, will it have the same amount of DNA as in parent cell in S phase? Students, remember, parent cell is what? 2 and 4C. Not the same. Does it have twice the amount of DNA in comparison to the haploid cell? Yes or no, students? Haploid cell is what? At the end, we have a haploid cell, right? At the end, haploid cell is what? NC. NC is the haploid cell. At the end, we have haploid cell is NC. Here we have NC condition. So does it have twice the amount of DNA? 2C and C. So can you see there is twice the amount of DNA? So this answer becomes right. So once you know the table, very well, you can answer such questions very easily. You have to only remember the table by practicing it enough and understand the concept over and over. I know the table can be overwhelming. The table can be like, too much. But trust me, once you master it, all of these questions becomes very easy. See, Same amount of DNA in comparison to haploid gamete? No. Haploid gamete is what? NC condition. So it does not have same. 4 times the amount of DNA in comparison to haploid? No. It does not have 4 times. It only has 2 times amount of DNA. So students, that right here is the entire question which we have the answer. Okay. Now students, with that being said, one question arises, all of you. We started with 40 picogram one cell. We are ending with 20 picogram. So it means that one diploid cell which had 40 picogram, it gave rise to 4 cell, right, which have 20 picogram each. How does it make sense? Students, what actually happened here was we had a diploid cell, it became haploid. This is what happened right now. Yes or no? Here to here is meiosis 1, here to here is meiosis 2. But how come from 40 picogram we are getting 4 cells of 20, 20, 20, 20, while this starting one has only 40 picogram DNA? How is it possible? Students, how is it possible? Yes, they exit after G1, Nancy. Nancy, they exit after G1. Students, do you have this question? See, here I told you, starting with 40 picogram here. In the end, we have 20 picogram 4 cells in total. How is it possible? All of you. Students, who can tell me? Who can tell me why? How do we have that? How is it even possible? How is it even possible, students? How is it even possible? How is it even possible? Students, answer is very simple. If you remember here, in your meiosis 1, in the S phase, the 40 became 80, right? So this 40 right now, it became 80 in the S phase. So this 80 is now divided into 20, 20, 20 and 20, and that's all, students. That's all. Students, do all of you understood? All of you understood the math here? This 40 picogram in the starting here, this 40 picogram in the starting here, it became 80 in the S phase. Now this 80 is now divided into 20, 20, 20, 20, and that's how you know the DNA count, students. Now students, here is a question for all of you. Here is a question to all of you. I want to answer in the chat. This is the mitosis, student. This is mitosis. That is your metaphase. Here's the question. Students, all of you start solving. Students, I'll give you notes, don't worry. In the description box, I'll give you, don't worry. Students, see this. Remember, I'll not even see the options, I'll teach you. Diakinesis. What happens? Termination of chiasmata. Packetine, recombination nodules crossing over recombinase enzyme. Zygotine, bivalent to tetrad formation. Leptotine, what happens? Chromosome becomes visible under light microscope. Synaptonemal complex formation. Zygotine. Completion or termination of chiasmata. That happens in the case of diakinesis. Chromosome looks like thin thread. That is your initial leptotine itself, right? Then students, appearance of recombination nodules. Packetine. Here's the question again, students. Here's the question again. Students, quick answer. I want quick answer. Following are the stages of cell cycle. Following are the stages of cell cycle, students. First, what do we have? We have G1S, G2, chirokinesis, cytokinesis. Okay, students. Okay, solve this question, students. Solve this question. Chromosome becomes gradually visible under light microscope during leptotin. Yes, makes sense. The beginning of leptotin stage is recognized by beginning of diploidin, students. Diploidin stage is recognized by dissolution of synaptic complex. Yes or no, students? Yes or no? All of you agree with me? Yes or no, students? Diploidin stage is recognized by dissolution of synaptic complex. If you go back to my notes right here, students, if you go back to my notes here, leptotin, zygotine, pacatine, here we have diplotin. Dissolution of synaptic complex, that is the first point here. So students, answer is obviously both are correct here. Both are right statement, both are correct statements here. Okay. See students, this chromosomes start pairing together to form synaptic complex, that is zygotine. Chromosome visible in light microscope, that is going to be leptotin. Four chromatids of bivalent chromosomes become distant and recombinant nodule. Recombination nodule, everything is pacatine. Dissolution of synaptic complex is going to be diplotin. Start solving, students. Start solving. See, very simple this is. Same repetition happens every single year. Same repetition happens. Okay. Process of recombination nodules, pacatine. Always, without even thinking. Without even thinking, pacatine you can tell. Okay. Which of the following stages meiosis involves division of centromere? Students, I told you, division of centromere happens where? It happens in anaphase of mitosis and anaphase 2 of meiosis. Right? So if it says following stages meiosis, division of centromere, students. Division of centromere happens in anaphase 2. Anaphase 2. Remember that. In anaphase 2, the centromere will split. Centromere will split there. Again, dissolution of synaptic complex, diploidin stage. Diploidin stage. Without even thinking, you can tell. Directly, you can go associate right now. Okay. I told students, during which stage of mitosis and meiosis, respectively, does the centromere splits? Anaphase of mitosis, anaphase 2 of meiosis. Okay. Students, correct order. Students, correct order. I'll give you 30 seconds to answer this question. 30 seconds to answer this question. Go. 30 seconds to answer this question right now. First we have what? Leptotin, zygotine, pacatine, diplotin, diakinesis. Yes or no? Let zebra play during the day. Let zebra play during the day. Yes or no? Which of the following never occurs in mitosis? Which of the following never occurs in mitosis? Never occurs in mitosis. It happens, does not happen. This pairing of homologous chromosomes, the synapses will only happen, students. Synapses will only happen in meiosis, not in mitosis. It's a meiosis thing. Appearance of recombination nodules on homologous chromosomes during meiosis characterizes the site of crossing over. I told students, site of crossing over. In prophase 1 of meiosis, chromosomes start pairing together and synapsis takes place. The process, students, synapsis, cytonymal complex formation, bivalent tetrad formation, all is zygotine. All is zygotine, students. Everything is zygotine. Beginning of diplotin is recognized by dissolution. Diplotin complex, okay. Formation of X-shaped mark called as chiasmata. Yes, makes sense. In oocytes of some vertebrates, diplotin can last for many years. Yes, it can last for many years. But is it explaining it? No, it is not explaining it. That is the answer. Oocytes of vertebrates gets arrested for years, months in. Again, where is it? Diplotin. In diplotin, it gets arrested for many years together. Okay, students, with that, we come to the end of today's class. Right, students, again, it's a reminder for all of you students in the description. If you can go right now, all of you right now only Once the class is over, tell us which state you are from, which city you are from, because that city will be choosing in this weekend will be coming there. Right now, students, I cannot tell any secret where we are coming actually, because we have gotten a lot of requests from one particular city. Okay, so we might be coming there along with other teachers, right? Now, students, let me tell you a small homework question right now. Small homework question. Okay, students, tell me in which stage of pro phase 1, right, crossing over takes place in the comment section. Students, crossing over takes place in which section? Okay, crossing over happens in which stage? In the comments. With that, students, with that we are done for today's class. We are done, students. Okay, with that, Kanul, I've told students, Kanul, understand Kanul, right? Here I told you, students, after meiosis 1, the resultant daughter nuclei will have N2C. The resultant daughter nuclei will have N2C after meiosis 1. Yes or no? So if you can notice here, students, after meiosis 1, here we will have N2C here, N2C. Now this N2C, right, N2C right now will have twice the amount of DNA in comparison to haploid. Yes, the haploid DNA is haploid DNA content is NC. Here we have N2C. So 2C is double. 2C is double. That's all. Okay, students. Thank you so much, students. I hope it is clear to all of you, students. The question is very simple. The question is that in which stage of prophase 1 does the crossing over takes place? Which stage in the comment section, students? Okay. Which stage do you think the crossing over takes place? Crossing over. The same stage we have recombinase enzyme, same stage bivalent, and we have recombination nodule also. That in the comment section, that in the comment section. Okay. Okay, students. So thank you so much, students. Bye bye, all of you. Take care. I will see you in the next class. Next class, I'll be teaching students again. Haploid is one chromosome. Yes, haploid doesn't have one chromosome. Haploid has Janani. Janani, understand. Haploid means it has one set of chromosomes. That is haploid. That is haploid. Haploid means it has one set of chromosomes, not one chromosome. One set means, let's say humans, we are diploid. It means we have 23 pair is there. In haploid, it only has 23 chromosomes. That is haploid condition. One set. Okay. Thank you so much, students. Thank you so much, students. I hope all of you. What to write for haploid definition? Told you, students. Haploid definition means a cell or organism which has one set of chromosomes. That is the meaning of haploid. Okay. Okay, students. Bye bye, all of you. Take care. Bye bye. Have a great day ahead, all of you. Boy, all of you. Students, let me know in the comment section how was the class. In comment section, Janani, Ansi, Kavya, Saka, Shamla, Nandini. Let us know in the comment section. Kasraj, right? How was the class?